Okay. Well, it's close to noon, so I guess I can say good afternoon, everyone, and welcome, certainly. This is a great day for us. I'm Mike Dake, Senior Vice President of Health Sciences, University of Arizona, and I'm really proud to welcome you to the first of our University of Arizona Health Science Research Showcases. Today's program is featuring 3 very interesting sessions focused on important work at the University of Arizona Health Sciences in pursuing, together with our partners, to tackle some of the most pressing health problems facing the world. I would say that our first session was fantastic. We talked about chronic pain and drug addiction, and it was just great. And I know this is going to be equal, if not superior. This is a great 2-session program on climate change. This session that we're going to enter in now is called Climate Proofing Food Security and Nutrition. And as we all know, clearly climate change is affecting our physical and mental health. The scale of the impact is vast, with millions of people around the world already facing the consequences of climate change daily. And at the University of Arizona, we're beginning to examine the main ways climate change affects human health. And we formed 2 working groups to bring together multidisciplinary experts from across the university and resources to address the effects of climate change on human health. In this session, you're going to hear from a variety of individuals who are part of these visionary group focusing on the impacts of climate change has on precision agriculture and nutrition. And I know you're going to be excited to learn, as I am, about the research we're doing on blue-green algae, known as spirulina, and the drought and heat-tolerant native Sonoran tepary bean as part of our program to help tackle global food insecurity. A few housekeeping items. The format of this session, we're going to start with sort of a overview and definition of problems, and then we'll have a series of speakers talk about some of our initiatives that are addressing this. You've heard a little about them. And then we're going to have, from one of our federal agencies, a representative talk about what's happening in terms of federal focus, funding, opportunities, future focus on climate change. Other than that, I will also say at 5 o'clock this afternoon, there's a reception for all of us up on the 13th floor. If you haven't been up there, it's a remarkable view. If the weather stays like this, you won't want to miss that. And then also the Wi-Fi is in the, if you're in the UA system, you're already connected, but if you're not, Wi-Fi is in the back. It is UADCCTR is the network, and the password is Wilbur #1. Also, I want to thank everyone who had a role in planning this. Obviously, when you plan a remote event, there's a lot of extra work that has to go on, and certainly we had a whole team, advanced team, working on it, including Melissa Colchado, Randy Wilming, Alex Dubois, Rachel Reinhart, and Brian Cunningham. So shout out to all of those. Also, thanks to all the audiovisual people. We're recording all of this, so if you want to refer to it later, we'll have that opportunity. And also a special welcome to our partners at CNRS who have joined us from Paris to come over. Many of we've met before, some I'm just meeting now, who are will be spending tomorrow with and exploring some of these opportunities that you'll be hearing about today. So now it's my pleasure to welcome our session's moderator, Dr. Floyd Ski Chilton. Dr. Chilton is widely recognized for his work on nutrition, and especially in the context of the human genome. He's been a pioneer in the areas of personalized or precision nutrition and wellness. He is director of the Precision Nutrition and Wellness Initiative, and we're honored to have Ski come up and guide us through this afternoon's session. Thank you.
Thank you so much, Dr. Dake, and thank you for your leadership. These are really, really important issues, and the fact that you have focused us on these important issues just speaks the world. I was just in Africa and in Ghana, and the world needs loving, it needs taking care of, and it needs leadership like yours. So thank you so much. I know when Dr. Dake came to us, I guess 6 or 9 months ago, and we began to address issues. One of the issues was really, how do we feed 10 billion people by the year 2050? Again, we need a 60% increase in food to do that. And if we don't, 3 billion people will die. And I know when we spoke to Dr. Dake about this, how in the world does one solve that problem? And it was almost ridiculous, the question, when we began to ask it. But believe it or not, when we began to go across the campus here at U of A, and one of the great things about being old like me is I know some of the smartest people in the world. So that allows me to bring in those people, and some of those people are in this room to try to solve this issue. So with that, I'll move on. And again, the name of, as Dr. Dake has said, is climate proofing and nutrition utilizing biofortified agriculture and alternative microbial protein sources. So this has been called the global syndemic. It's climate change, undernutrition, and obesity. And they're all attacking us at the same time. So the fundamental challenge is, how do we produce enough healthy food to feed a growing population without destroying the earth in the face of climate change? It's a really important question, and it's an extraordinarily difficult challenge. As I said, food production will need to grow by 60% to feed the 10 billion people on this planet by 2050. 690 million people experience undernutrition, with 48 million children experiencing wasting, another 147 children experiencing stunning. Again, food production, we're trying to do this in the face of food production causing the most environmental damage of anything we do. So we need to produce more without damaging more, with damaging less. So we must move to plant-based diets and again. So we've been here before. Again, the great thing about being old is you remember. And I remember this book by Paul Ehrlich called The Population Bomb. And the population bomb is essentially the bomb that we're experiencing now. Only he published this in 1969, and basically he said, unless, he didn't say unless, he was very negative. He said, we're going to not be able to feed this planet, and billions of people will starve if population keeps increasing. Now, his prediction did not come true because of the first Green Revolution, and that was largely responsible by a person named Norm Borlaug. He was an agronomist, and he led a worldwide initiative that extensively increased agricultural production. He developed semi-dwarf, high-yield, disease-resistant wheat varieties and other varieties, and he also introduced the widespread use of fertilizer. So that produced the first Green Revolution, and it saved billions of people's lives. Okay, we've pushed that as far as it can go now. What do we do now? Next slide. Oh, I'm sorry, I'm controlling that. So the Green Revolution, in addition to producing more food, it caused a whole variety of problems. And I'm not going to read all these. I'll let you read all these. But again, the widespread use of chemical fertilizers and pesticides, soil degradation, water depletion, large loss of biodiversity, adverse effects on indigenous crops, and really adverse effects on health around the world, including obesity. For some reason, this is not going forward. Just a second. I can certainly use a remote. No, I can't. If someone could walk up here real quickly and help me,
Where's the, let's see this.
Okay. Okay. So why the University of Arizona? We reside in the Sonoran Desert. The University of Arizona leads the world in agricultural and nutritional innovation. And we like to say, and I think Duke may have come up with this, we like to say that we are preparing for tomorrow's climate today. This is what the rest of the world will look like in 20 years. So how do we innovate? I'm not going to go into this slide extensively, but I have started 5 startup companies. And one thing I've realized is perhaps the greatest innovation in any startup is the vertical integration of all the technologies. Unless you get from the beginning to the end, you don't win. So we'll be focusing on vertical integration. And this is today's agenda. I don't need to go over it. It's my job now to introduce one of my friends, Dr. Duke Pauly. Duke is an assistant professor. He focuses on the genetic basis of stress adaptive traits in crops. His skills and experience span a wide array of crops, including growing and evaluating lettuce underneath the UA field scanner, which is the largest outdoor phenotyping robot in the world. In addition, Duke and I spent 2 weeks at the bottom of the Grand Canyon together, so we know each other quite well, and I'd like to welcome Duke to the stage at this time.
All right. Well, good afternoon, everybody, and thank you for being here. Ski, thank you for that wonderful introduction. I'd also like to thank Dr. Dake and his team for organizing the whole event. I know, like you said, doing this remotely takes a ton of effort, so very appreciative of that. Today, I would like to talk to you about the tepary bean. This is a little-known desert plant that has tremendous potential. And I know you're kind of like talking about beans, but if you'll allow me to tell you the story of this specific desert plant, I think it highlights what a climate resilient crop actually looks like. And more importantly, I think it shows what the potential looks like as we think about addressing food insecurity. And finally, the additional presentation by my colleague, Dr. Arnold, who will follow this, it might challenge your perception of what is possible with arid land agriculture. I would also like to mention that the work that we'll be discussing today falls into a recently launched center at the University of Arizona called Agroecosystem Research in the Desert. And this center was started largely addressing the question Ski just posed, is how do we feed our planet without destroying it? And so when Dr. Dake came to us, it was this challenge of how do you provide access to food in the face of climate change while promoting human health? Certainly no small task. But what's unique about the University of Arizona is that we have this breadth of expertise from health sciences to Ski's Precision Nutrition Center to plant biology. And we have this unique ability to leverage our environment to begin addressing fundamental questions facing society, and specifically some of the issues that agriculture is facing. And so what are these issues? So these are the issues that we read about in the newspaper on nearly a constant basis. Obviously, in the last 10 days, it's been dominated by the conflict in the Middle East, but we are seeing the increased prevalence, intensity of drought, heat, and a water shortage. So with respect to drought, more frequent, increased severity, larger geographical area impacted. Areas like the Midwest and the Northern Great Plains, which traditionally have never had early season drought, in the last 3 years have been impacted 2 out of the 3. We talk about heat, more intense heat events, higher nighttime temperatures. This is critical. This is what allows organisms to recover. Plants, animals, humans, we all operate the same. And then increased duration of heat events. I don't know about you, but I had never heard about a heat dome until 3 years ago. And I study heat and drought research, so this was certainly a shock to me. More locally in Arizona, we just had one of the hottest summers on record. 31 consecutive days above 110 degrees Fahrenheit. This type of heat is unheard of, and it's only increasing in terms of its severity and impact. And then finally, a water shortage. This is becoming a critical resource throughout the world. We know there's less of it, access is limited, and there's extremely fierce competition. Right now, a town in California is tearing itself apart, community versus growers, in terms of accessing and pumping groundwater. So we know these threats to agriculture exist, and they're only getting worse. So what is one of the solutions to this? And this is this concept of climate-proofing crops to safeguard calories. And so we know that genetic and crop engineering, we've seen this type of technology, it exists. It does have the potential to provide solutions. However, we're just not there yet. The real idea of an engineered crop to withgo abiotic stress is probably 15 to 20 years out. Additionally, engineered crops likely won't make it to where they're needed. Licensing fees are too high to really get these crops into third world countries where they're truly needed. So we need solutions right now that can kind of bridge this gap. And we need to leverage our technology and the science that we have now to begin coming up with solutions. So I'll be talking about this native plant tepary bean, and Dr. Arnold will be talking about soil microbes. And the combination of these two really offered a new paradigm in arid land agriculture. So what is the tepary bean? I'm sure most people have not heard about this plant. It's native to the Sonoran Desert. It's one of the true stars of the Southwest. It's been cultivated for 5,000 years. And really, it sustained an ancient civilization, spanning from, Arizona into Mexico. And that's because it's a high protein source. Additionally, because of where it's native to and adapted to, it's already highly heat and drought tolerant. And this really makes it an ideal crop as we think about what future climate scenarios look like. And finally, it's highly nutritious. It's often cited as one of the healthiest legumes out there, even though if you Google it, it won't show up because it's not that well known. It has a high protein content as well as high soluble fiber. So what is it about these bean plants that make it so resilient in the desert? It has a host of tricks up its sleeves, including the plant properties itself. So the shape of the leaf is extremely teardropped. Why this matters is it radiates excess heat away from the plant itself, reducing stress. Positioning of the flowers in the pods. You can see in this photo, the pods and flowers on this plant actually form in the middle of the plant by the ground where it's coolest. So the plant basically creates a microclimate for its reproductive structures. It has dynamic positioning of the leaves. It's one of the few crop plants that can actually turn its leaves on edge during the middle of the day so it's not absorbing sunlight. These tricks we don't see in other types of crop plants. Has rooting properties. It can put down roots steep and deep very fast. It can chase water. In terms of water requirements, it's pretty minimal. I have the distinct honor of being one of the few people that have killed a tepary bean crop by overwatering it because I was scared, because I was like, surely I need to put more in the profile. I was wrong. It thrives on sandy and well-drained soils, the types of soils that you find in deserts, or the types of soil that start to develop under aridification scenarios. And finally, again, the plant responds very rapidly to water when it has excess, and it really knows how to sense its environment. Agronomically, it's very flexible. It's a true short season crop, anywhere from 60 to 90 days from planting to harvesting. If you're a grower, that means you could double crop it, or you could add it in a rotation to make more of a return on investment. I've already talked about its nutritional properties, but the plant biomass itself can be used as forage or feed for livestock. And then finally, it's a true crop that doesn't require a lot. Because it's a legume, you don't have to add fertilizer. Again, the watering requirement, minimal at best. So what work is going on at the University of Arizona with respect to taproot bean? So right now, this plant has largely been an orphan crop, meaning that it hasn't been invested in heavily. And so at UA, what we're presently doing is we're starting to modernize the crop. So we're getting the genetic information, the genome sequences, as well as the phenotypic information from controlled field environment studies. So what we're doing is basically growing these large panels, 342 different varieties of taproot bean. We're growing them in controlled irrigation scenarios where we have a well water to control, if you will, and then a drought scenario where we truly stress the plant to see what it's capable of. And what we want to do is we want to measure their growth rates in response to heat and drought, not just their final yield. We don't always care about that. We want to know how dynamic the plant itself is to the environment. And then finally, we're only beginning to really kind of ask the question is, what do the nutritional profile of these beans look like in our unique environment where it is so hot, dry, and limited resources? So with respect to the actual nutrition of the bean, it really is kind of a superfood, respectively, for a bean plant. It has extremely high protein content. Legumes are already recognized as being, a high source of plant-based protein. This bean actually tops the list. Again, you won't, if you Google it, probably won't show up, but primary literature kind of bears this out every time. Under high input crop scenarios, so this is when you're adding phosphorus, potassium, other micronutrients to the soil, you can achieve up to 30% plant protein. To give you an idea, corn, wheat, you're up to 10 to 15% range. Micronutrients. So because this plant is so good at dealing with stress, it's developed novel biochemical pathways and processes that actually result in the bean itself having more micronutrients compared to other crops like common bean, like navy beans, pinto. Fiber, our good friend fiber. It's essential in terms of promoting human health. This bean has a high amount of soluble fiber. It's slow digesting, so it creates more sustained energy, a feeling of fullness. It prevents overeating. Additionally, we know that fiber is extremely important in controlling cholesterol, blood sugar with respect to diabetes, and human microbiome health. And as we continue to learn more about gut health, we know that impacts every part of human well-being. In terms of fat content, it has a very low fat content. The fat that it does have is very healthy. It's all unsaturated. There's very little saturated fat in these beans. And this is what's really remarkable about this bean and why it was such a key crop in the southwestern ancestral peoples, is because it achieves so much in terms of providing protein. If you've been to the desert, there's not a lot of wild game or other sources of protein to capture or eat. And this is why they relied so heavily on this bean. Between that, its drought resiliency, this really sets the tone for what a drought resilient crop, a climate proof crop looks like. But if we're thinking about what we can do to improve taproot bean, there's one thing that we drastically need, and that's genetic diversity. So in a current study, first let me just start off, why do we care about genetic diversity? Genetic diversity is the building block of plant improvement. Basically, if you want to have the tallest plant or the most nutritious plant or the most heat tolerant, you have to have a collection that has those lines that are outlying performers, because that means they have the genes that are basically controlling those traits. So our first step with this project was to undertake whole genome low-pass sequencing of 342 of these taproot lines, resulting in about 6 million markers to really look at what the genetic diversity is. So in this graph, we basically have the 4 types of taproot bean: cultivated, wild, breeding lines, as well as the weedy type. These are really, truly wild. This is what these look like on the side of a road. And then the size of the circle indicates the number of individuals. So the bigger the circle, it means we've collected more lines from there. And then you can see the proportion breakdown. What this graph summarizes for us, though, is that our population that we're studying basically really captures all of the extent variation that's present in this crop throughout Mexico and the U.S. Southwest. And this is critical because if we have any chance of improving it, we have to have access to all this diversity. The other thing I wanted to mention was the dynamic response to the environment. So what I'm showing here, so we use UAVs, drones, to measure how the plant grows and responds. And what I'm showing here is a trait called green leaf index. So not only does this track the size of the plant, but it tracks how green it is, because again, this plant has a remarkable ability to say, hey, I'm running out of water. I don't need to invest in more leafy biomass. I need to take that water and use it in my reproductive structures to make beans. So on the right-hand side, we basically see the trajectory of the plant growing with irrigation. It's a continued uptick in the slope until finally it reaches its peak. On the left-hand side, though, we see something interesting going on. So we start the trial. We withhold water. We see a severe collapse in the growth rate, the greenness of these plants. We supply irrigation water. Within a matter of 5 days, the plant has recovered to almost the controlled conditions. This does not happen in other crop plants. Wheat, cotton, corn, nothing can respond this fast. And we can repeat the same pattern. We withhold water again, the greenness goes down. As soon as we bring the water back, it bounces right back up to control levels. On the right-hand side, you can see the yield for this entire experiment. Normally, you would expect to see a pretty significant difference between, a drought treatment and a control, especially when you just went through the hottest month on record in Arizona. We don't really see that. In fact, we had lines that performed better under the drought conditions. Again, highlighting that this plant knows how much water it needs, and it knows how to use that water most effectively in terms of producing seed yield. So this is really remarkable to me, and the fact that any crop plant has kind of figured this out, but more importantly, that any crop plant can be this in tune to its own environment. So with the support of Dr. Dake, we were able to take a subset of these lines, only 48, and actually look at the nutritional profiles. And this is what gets really exciting for us. So of these 48 lines, we see in the first left-hand graph of seed yield, there's no difference. They yield about the same. However, when we look at the percent protein, we see that under drought conditions, the plant actually makes more protein. It goes up to about 21% on average, versus under control conditions, it's at 19. We see this in other grain crops. We'll see the protein levels go up, but the yield decreases, and that's because the plant, like in wheat or maize, it's producing less starch. So it's not that the plant's producing more protein, it's that there's less starch. We don't see that with tepary bean. When we look at the fat, minor differences. When we look at the micronutrients, no differences. So what this really highlights is the ability of this plant to be a true desert superstar with respect to providing nutrition under really tough conditions. Again, just want to highlight that this was done during this heat wave that we just had this summer, and we still had these types of results. So this is what's really exciting about alternative crops, is there's something that we have failed to look at, but it has this huge potential. So I'd like to spend just the last 2 minutes kind of discussing, why agriculture? When we talk about climate health science, how does agriculture fit in that, picture? Well, we know that food insecurity is a huge multiplier, right? It starts at the individual level, going to the family, to the community, and finally to the socioeconomic and basically, country level scale. We see mass migrations when people run out of food. The other big thing is that agriculture and related activities employ a quarter of the world's population. That's a lot of people that rely on agriculture for their income, for food, and for their livelihoods. And so I'm glad that Ski mentioned the Green Revolution. So why is providing enhanced seeds so important? It's because once you have this, developed variety, the plant itself replicates the technology. There's no more human intervention needed. You just grow the plant. But more powerfully is, I can take that harvested seed, I can give it to Ski. He grows it. He gives it to Mark in the back of the room, right? Mark doesn't have to come to me anymore to get the technology. It's being distributed broadly. And because of this, and this is why the Green Revolution was so successful, is because the seed got to where it was needed. And this is why improved plants have so much promise with respect to agriculture, improving climate security. And then finally, sustainable agricultural practices. For subsistence farming, this is largely already done. We're not using diesel, we're not using big tractors, things like that. And more importantly, there's been this resurgence of interest in this. Now it's called regenerative agriculture. But it really highlights that if we take care of the land itself provides an ecosystem service for us. Improve soil health, so carbon storage, water retention, dust mitigation. Some of the things that are already impacting our society can be accomplished with just through, improved agricultural practices. The next question would be, why tepary bean? Why beans in general, really? The biggest thing is that there's a lot of tools in the toolbox of fighting climate change and human nutrition and food security. But I would argue that tepary bean is probably one of the first tools that should be used. Why is that? It's because nothing new is required. This is truly a plug and play. There's no new tools needed. There's no new cultivation methods. And this is really the power of the plant itself, is that it plugs into what's already existing. So the technology to harvest, to package seed, to distribute, it's already in place. I've been hopefully convinced you that it's climate resilient. Truly, among any other crop plant that we grow or know of conventionally, it's at the top of the list. Broad adaptation. Growing outside the U.S., not a problem. It's grown in Africa presently, as well as South America and India. That's another benefit, because dry beans are already largely consumed in areas that are threatened by food insecurity. And, integrating tepary into these diets would not be a large hurdle. This is one of the key things, is that food is culture. And when you try to bring new solutions to people, it's also convincing them to eat something new that really isn't part of their natural diet. Finally, it's a low input crop. We already know this. In terms of fertilizer, water, doesn't need a lot of human intervention. It's quick growing, so there's a chance of doing double cropping, high rate of return on investment. We know it's high in protein. Yes, animal protein is, high quality, and there's a lot of it, but it's access limited. Animal protein in most countries is a luxury. We also know that animal-based protein is resource intensive and really has a negative impact on the environment. And we know that plant-based protein is going to be something that we have to continue to invest in. And finally, it has built-in food security. So if it doesn't look like you're going to make a bean crop, that's fine. You can let your animals graze it. Don't have animals, even better. You can plow it back into the earth, and it forms a natural fertilizer, right? Because it's a legume, it's fixing atmospheric nitrogen, so now you've just fertilized for free. So you could follow with a higher value crop or fix in something else. So this is why tepary is such a powerful plant. Why it hasn't been done yet is because really we haven't had access to the germplasm, the genomic resources to really study, understand, and improve the plant. But now we do. And that's why UA is kind of one of the leaders in this space, and why this plant has so much potential when we think about climate change. So finally, I would just like to leave you with a couple questions. So we already know that tepary is naturally a desert master, but is there room for improvement? We talked about genetic improvement, but we're not quite there on that same time scale. So given the rate of change in our environment, we have to come up with some ways to enhance its profile, its ability to grow and respond to drought stress, its nutrient profile. So what kind of solutions are out there? And I would like to leave you with that as my colleague, Dr. Arnold, speaks next, but just to kind of set that stage of what's possible. And, briefly, and it always takes a ton of people. It's just a very short list. But with that, I thank you for your time. And if there's any questions, be happy to take them. Thank you.
We're going to do questions at the end.
Perfect.
It's mine.
Oh, yes. I just set it down there.
Thank you, Dr. Pauly. One of the great things about this, and one of the kind of maybe not so great things, is we've organized it, but we've not heard the talks. And hearing the talks, we're talking about another green revolution. We really are thinking big here, and it just so excites me. Thank you, Duke. That was fun. That was fun. Well, speaking of fun, I want to next introduce Dr. Betsy Arnold. I often say that Betsy may be the smartest person I know at the U of A, and at the same time, the nicest person I know at the U of A. And the same time, I keep finding that she's connected to the Waltons and all that. So she may be the most connected person of the U of A. But Betsy is a great researcher. She has an interest in ecology, evolution, especially with symbiotic microbes and plants. Again, her team collaborates on a wide variety of questions: conservation, agricultural innovation, pharmaceutical drug development, and biodiversity discovery in diverse settings. And I just can't wait to hear your talk, Betsy. So come on up.
Well, now that Ski has set some really reasonable expectations for me. Hi, everyone. I'm Betsy Arnold, and I just really want to thank you all for being here today. It's really a pleasure to see familiar faces and many new faces, too. I'm really honored to have a chance to talk about some of the work that Duke and I have been able to put together in these last months with support from you, Dr. Dake, so we really appreciate that. And I'm just thrilled to share some new data and new perspectives with everyone, so thank you. So to start, what I'd like to do is transition us from the green world around us and to recognize that every time we see a plant, we're seeing a microbial community. And when we think of that microbial community, we're actually witnessing a biodiversity microcosm that has resulted from the power of ecology, the power of evolutionary biology, and the power of biodiversity. And so when I look at a crop field, what I'm seeing is both the product that we're after, that food product, that fertilizer product, as you can plow chaparral bean into the ground, that fodder, forage, or fuel. But I'm also seeing a tremendous wealth of biodiversity, an untapped potential that's in the soil, that's in those plants, and we'll talk about that today. In particular, we're going to be talking about plant microbiomes and how we can use them as a lens to unlock what I like to call 1 billion years of R&D. That is the long evolutionary history between microorganisms and the photosynthetic hosts with which they live. And we'll talk about how we can translate that to climate-proof crops. So I wanted to start just with the simple concept. We've heard already from Ski and from Duke the challenges that we face in agriculture. And arguably, we can imagine that in the simplest way, agriculture today is going to rest on 3 pillars. We have the history of plant breeding, coming up with varieties that are resilient and productive. We have plant modifications, our current world of modifying that genetic germplasm. And then we have inputs of fertilizer, inputs of water, inputs of human labor. And all of those things support our food system today and are vital to our existence. Now, 3 pillars can be great, but if that set of 3 pillars is on uneven ground or shifting ground, then we find ourselves suddenly realizing that it's quite easy to topple our food system. What kind of challenges do we have that underlie that uncertain ground? So first of all, limited resources and inequitable distribution across our planet. Second, acceptance, discomfort with the concept of GMOs. Third, time, the rapidity of change that we are facing. And then finally, that existential threat of climate change. And so when we're starting to think about what tools we might have, and I thought that Duke ended on a really good note, it's a great example of an ideal crop, but where can we take it next? This is where we can start to think about the world of plant microbiomes. And this is one of my favorite photos from an area near Tucson. And we can look at this abandoned agricultural field and see nothing but dead plants and one little relative of tomatoes, potatoes, eggplants flourishing in that space. Now we can ask, is that something genetic about that plant, or is that something symbiotic about that plant? Well, it turns out that when we start to shift, just as we have done in medicine, from thinking about microbes only as a source of disease, and instead to recognizing that often when we see disease, that's a world of dysbiosis, we want to be thinking about microbiomes as the tools for symbiosis, that is, for the flourishing of a host organism. In medicine, that's increasingly important, and it's certainly important when we look at the landscape around us. And so arguably, the world of plant microbiomes is a world of paradigm shift, where we're starting to think about how do microbes serve a positive role in the plants all around us. And those microbes are tremendously diverse. When we start looking inside plants, you'd be shocked. I had a delicious salad last night and reflected on the microbial diversity I was happily consuming. These are just a few cultures from our lab showing a diversity of some of the fungi and bacteria that can be isolated readily. But what's exciting right now is that we're not constrained only to isolating microbes in culture. We have the power of the omics revolution, and we're in the position to use high throughput tools to very rapidly capture a picture not only of who is there, but what their evolutionary history might be, what their genetic diversity might be, and how they can be used going forward. So I always equate looking into a plant for microbiome as sort of getting the world's best birthday present, because you never know what's going to be inside that box. But once you can start predicting it, you know you're getting the most exciting gift ever, which is this gift of biodiversity. So with this in mind, why do I call this a billion years of R&D? Because over the evolutionary history of plants, they have coexisted with microbes in every environment. Indeed, to be a plant today is to be a microbially symbiotic organism. We find microbes both on plant tissues and within those living tissues themselves, and in the soil that surrounds them. The particular microbes that you'll find associated with any given plant are going to be influenced by plant traits, by chemistry of those tissues, and the environment in which that plant is growing. We now know that very much like our own microbiomes influence so much of our health, these plant microbiomes are responsible for major elements of plant growth, plant productivity, and food safety. And so needless to say, a holistic or systems-based approach to thinking about agriculture needs to incorporate these hidden components, too small to see, but incredibly important. And just how important can they be? Well, what we can see is that we might take genetically identical seeds and treat them with a diversity of microbes that we've isolated from the field. And what we'll see is tremendous and rapid differentiation of their tissues, changes in root architecture, changes in germination success, changes in drought tolerance. And what's amazing, of course, is that every single plant harboring a microbiome means that those microbiomes are all around us. So I've been very fortunate over the past 20 years to work throughout the world, looking at plant microbiomes, sometimes in places as plant-rich as the jungles of Central America, other times in environments that might not bring to mind biodiversity, an agricultural field. Well, what's interesting is that when we think about the power of green revolution, what we have seen is an incredible transformation for human well-being. But the challenge that we face is degradation of those soils, okay? And also the challenge that modern agricultural practices typically work against symbiosis. If we imagine, for example, the human diet, supersized on nothing but I don't know, my favorite cheeseburgers, right? We would find ourselves in a period of dysbiosis. And so if we equate that to fertilizer inputs and water inputs, massive contributions to plant health in the short term, we see immeasurable costs to plant health in the long term. And so as we move from a world of sort of supersize me in agriculture, we need to be thinking about the mini aspect, the tiny aspect, the microbes down below. And so at the University of Arizona, we have our living laboratory, Arid Land Agriculture Today, and we really see that as the toolkit for understanding tomorrow's climate. As Duke outlined, we're in a position where in field conditions, at large scale, we can grow diversity panels of plants in shared environments. If you're like me, you can take advantage of all the work that Duke and his team do to go in and look at the microbes that are there. And in particular, we can study the microbes that are recruited to crops under severe stress. And what's powerful is that we can pick out those high-performing crops. Okay? You are in an environment where no crop in any right mind should be successful, yet you're performing, you're yielding products. What is your microbiome? So that's the approach that we take. And over these decades, we've generated the world's largest collection of plant symbiotic endophytic fungi and bacteria. These are those microbes that live inside these healthy plant tissues. And we also have the largest collection now, in part with Dr. Dake's help, in arid land microbiome data more broadly. So I'd like to maybe leverage what Duke just talked about and revisit tepary bean for a minute. So it turns out that tepary bean, as a desert-adapted superfood, actually has an additional element in its arsenal. In addition to all of the tremendous traits that Duke outlined, it actually has an exceptionally powerful microbial toolkit that with Dr. Dake's support, we've been able to examine over these past few weeks. Now, what I'd like to do is just show you a few data points from our recent work. And the first message I'd like to convey is that when we compare high-performing tepary bean against lower-performing tepary bean, we see a distinctive microbiome even under shared conditions. So each little point on this graph represents hundreds of thousands of DNA sequences. And when we see these different colors, here we see the microbiome space of our high-performing plants. Here we see quite distinct the microbiome space of our low-performing plants. So in the same environment, the winners are picking winners. Okay? Now the second thing that I want to convey is that in the case of tepary bean, we see that tepary bean very rapidly shifts its microbiome when it's under stress. Here, what we can see is a transition from high water to low water in the same field. Okay? Dramatic change in that microbial association. And then finally, we see that high-performing tepary bean harbors a very distinctive and novel microbiome. And that's in its roots, in the brown, in its leaves in the green, and even in the beans. So as we start thinking about micronutrition, let's recognize that tepary bean itself, the bean product, is rich in bacteria, rich in fungi, completely understudied, and yet almost certainly important in our nutritional cycle. Okay? So these data are, I can say, hot off the press. And there's one other element that I just want to highlight about our work so far, and that is that high-performing tepary bean recruits many different kinds of symbionts. Those different kinds of symbionts have complementary function. So I just wanted to share, this is some microscopy that I did where we're looking at a group called dark septate endophytes. They occur in roots. And that when we start culturing the bacteria, knowing what's there, we can adjust how we culture them to get more into our biodiversity collection. We see a tremendous richness of gram-positive bacteria in the roots. These are ideal for seed coatings, basically to treat seeds, to plant those seeds so they are successful in turn. Okay, so if you were to summarize these findings in maybe just one sentence, what we can say is that the highest performing plants in tepary bean, as an example, are what we refer to as symbiotically competent. They are good at symbiosis. They are good at making these partnerships. Our high performers are distinct in their microbiomes. High performers rapidly and adeptly will shift their symbiotic partners under stress. High performers have more types of symbioses. And we think that there's some real potential here for micronutrition and pre or probiotic potential of the beans themselves. Okay. I also just want to highlight that this is now a rapid process to be able to capture the microbiome associated with a given crop or host. This is now on the order of a 1-week turnaround. So we can go from field to data set in real time. And that's something that we are uniquely poised to do at the University of Arizona. We also now have a really streamlined workflow where we go in about 6 weeks for a culture-based approach to capture the biobank of microbes. Now, we've talked a lot about tepary bean this morning, but of course it's not the only crop. And I want to note that these core traits that we've just talked about of being symbiotically competent, especially for high performers, this is a pattern that we're seeing in all the crops that we've looked at under arid land conditions, for cotton, for sorghum, and where we've been able to do it with lettuce as well. What I also like is that these fundamental studies enable some new innovations. For example, there's a lot of interest in how do we transfer the desirable phenotype or traits of a crop from one plant to another instantaneously. So one way that we can do that is to move the entire microbiome, not shift just one or two members. And so what we're seeing in this simple example from a student in my lab is that we're seeing a low-performing line of sorghum that received a low-performing microbiome, and a high-performing microbiome moving on to a low performer. We see differences in water use, we see differences in growth. And so we can actually transform a low performer to a high performer. We also then can start having integrated approaches as we've developed as our Lettuce One Health concept here, where we're taking into account our root and soil microbiome, our leaf microbiome, how those influence plant resilience, plant health, crop quality, environmental sustainability, and ultimately human health. So now the currency that we have for all of that is genomics, is DNA sequences, is transcriptomics. So in my last few minutes, I just want to talk a little bit about how do we source beneficial microbes. And this is a biodiversity and informatics initiative at the University of Arizona. Our approach is that we focus first on crops and their wild relatives. So Arizona is a biodiversity hotspot. We have plants representing close relatives of many of our crop plants occurring naturally in degraded soils, often in very hot and dry conditions with no inputs. When those plants, like that photo I showed you early on, are flourishing in those landscapes, what a great place to go look for microbiomes. So we focus on high-performing crops under stress, as I've told you with our tepary bean example. We're interested in crops' point of origin. How did they emerge with their microbiomes from where they evolved? Undesirable weeds and wild plants are really terrific sources and often very productive for us. And then finally, we're interested in ancient and resilient plants that flourish in hot conditions. And so here we're seeing a plant that no one would mistake as a food plant, but as it flourishes in the desert, it has established partnerships with a desirable panel of microbes. This, of course, is not an Arizona-only endeavor in that the kind of work we're talking about is very amenable to international partnerships. We have a proof of concept now where we're looking at sorghum microbiomes in areas that are ostensibly quite similar in terms of climate in Kenya and Arizona. It turns out that the microbes you find in those two environments are very different taxonomically, yet functionally very similar. So what rules can we start to draw to understand how those things work? When we're working overseas, we have a terrific opportunity for capacity building. It turns out that many of the technologies we use are incredibly exciting for students. We've hosted workshops in many of the countries where we've worked, promoting basically the standard techniques of sterile technique, how to isolate microbes, how to sequence DNA. And that's been really high yielding in terms of connections. Because the microbes differ as we move around the world, it creates some potential and some challenges. So in biodiversity, we are governed by the Nagoya Protocol. This has to do with the rules for using biotic resources from developing countries. If you can imagine in Kenya building its own biobank, its own indigenous microbe collection, that's something that we've now done in a variety of countries with a number of different partnerships. And this adds a number of value-added benefits. So for example, we're interested in pharmaceutical drug discovery. This is a model that has been employed with my collaborators in Panama and has led to PhD programs, capacity building, and drug discovery. So these international partnerships then open the door to international development through a microbial lens. Okay. At the University of Arizona, we lead in that biobank development, and our workflows are easily deployable. So for a relatively low cost, it's quite straightforward now to generate such a collection. So how do we tap the challenges and the promise of plant microbiomes in our changing world? Well, I think we have some challenges before us. For example, regulatory challenges are important. At the moment, for example, there is a tradition in the United States for there to be an approval of a microbe as a crop additive only if that microbe is identified, only if it's not on a list of select agents. However, many microbes out there are not yet identified. They don't have a traditional Latin name, yet their benefits are very clear. So what we need is a fusion of new thinking, systematics, knowledge, evolutionary biology, and regulatory to come together and assist in a new learning curve for some of our regulatory agencies. The second is that there has been a history of challenges in seed application technologies. This is changing. Now a focus on gram-positive bacteria has made seed treatments much more tractable than before. Moving toward a systems approach in agriculture, understanding the benefits of these cryptic microbes, and then finally linking these microbiomes to phenomics, or the study of plant phenotypes at a large scale. The urgency is tremendous as well. So we're talking about losses of biodiversity that are unprecedented across our planet right now. We don't know how many microbes are being lost on any given day. We don't know how the loss of a single forest tree might compromise our ability to solve human problems going forward. I'm interested increasingly in a microbial lens to conservation, and I think the tools are in place now for something that we've never been able to do before, but to make that cryptic biodiversity an incredible important area of focus. And then finally, of course, we have the existential challenges that are facing us. Last, I'll just note that right now, work at scale is relatively closed source. There are individual scientists like me who are out looking at microbiomes and how they can solve problems. There are small consortia, but nothing at a national or global scale. And so this is an open space, one that I think could be transformative for human well-being. And where it's happening at large scale, for example, in seed treatments or in modifying agriculture, is entirely in the hands of private industry. It's closed information. There is no moonshot where people are coming together to find this new way forward. So if that's something I could change about the world around us, it would be to discover those microbes, to conserve them, to understand what they do, and to work at a national or international level to get a handle on that cryptic, genetic, chemical, genomic, and functional biodiversity that is too small to be seen but powerful in impact. So with that, I'll just close by saying that at the University of Arizona, we have the confluence of our health sciences leadership. We have our microbiome biobank that I administer, the largest of its kind. We have our arid center. We have the arid land agriculture, and we have the technologies to move these endeavors forward. So with that, I'll go ahead and stop for today. I publish as A. Elizabeth Arnold, an international woman of mystery, I guess. But you can call me Betsy. And I just really want to thank you so much for your time. Thank you.
And now you can see why I was excited. She's amazing. She really is. Thank you, Betsy. I love the way you walk around. It reminds me of a church or something. I feel like I should say, can I hear an amen? That was really nice. Thank you. Well, the next speaker is actually a hero of mine. Actually, both of these gentlemen in the corner are heroes of mine. They have been all over Africa and been all over the world and really impacted the lives of pregnant mothers and severely malnutrition children throughout the world. Mark is going to speak with us, Mark Maneri. Again, one of the great things about being old, as I said, is I get to know people. And Mark and I just spent the last week in Ghana. So we've been together for a while. Mark is probably, undoubtedly, the authority in the entire world on childhood malnutrition, and especially childhood malnutrition in difficult places. I'm not even going to read his bio. Mark's a hero of mine. Mark has carried out 30 to 40, again, I have no idea, clinical trials in Africa in some of the most difficult places in the world, and he's published them in the best journals in the world, the best clinical journals in the world. Sitting right next to him is Tom Brenna, who've combined putting omega-3 fatty acids for brain development and the combination of ready-to-use therapeutic foods and ready-to-use food supplements, again, throughout the world. So welcome, Mark. Thank you. Thank you for coming.
Thank you, Ski. And now for the hard part, figure out how this thing works here. Great. So what I want to talk about is arguably the biggest nutritional problem that the world is facing now, which is dietary protein quality. Very briefly, I'm a confusing character to you, but the best way to think of me as the CEO of a not-for-profit that operates in 4 countries in Africa. I have about 200 people that work for me. About half of them work delivering clinical service, and the other half work producing food. And then I want to bring up some hope, which I believe is in spirulina and mycoprotein. So this is about a little bit of basic biology for everybody, but proteins are these bending, beautifully shaped chains that are made up of 20 building blocks. And the building blocks are universal. Every one of us, every living thing is made up of these building blocks. And the shape of the chains is what gives it function. So every functional part of biology is a protein. And then when we think about what we do as people to get our protein in our bodies is we have a digestive system where we basically mechanically crush things in the mouth. We drop it into a high acid environment in the stomach where everything gets stretched out and certain kinds of chemical bonds are broken. We move it to a high basic environment in the intestine where enzymes then cleave the food into little individual pieces, to these building blocks, if you will, in terms of protein. And then we have a middle part of our intestine where we turn it over to the microbes when we're done with whatever. We're done with what we can do with it. So the first message I want to get across here is protein's a very old-fashioned word. It means something with having nitrogen in it, but we really should think of it as 20 different nutrients. And the two that are usually in tightest economy are some things called lysine and methionine. Now, how much protein is needed? Well, it depends on who you are and what you do. So these bar graphs over here, and we'll see which ones I'm looking at there. The black bars, these are for countries in Africa, for different, and show if you have a fairly sedentary life and a good diet, what your protein requirement might be. But if you have an infection, it goes up over twofold. And lots of people in the developing world have a lot more infections. If you're growing or doing lots of physical labor, it also goes up. So the protein requirements for people in the least developed parts of the world is actually higher than it is for those of us here in this room. And then on the left here, you see these lines, and these are the darkest line at the bottom is adequacy, and you can see the places of the world are here, and Africa's here, and North America's around the corner here, and the greatest deficits are in the poorest societies. Okay, so this is why I say this is the nutritional challenge of the 21st century. We figured out how to get micronutrients into things with food fortification, with micronutrient supplements pretty well at this point in time, and now it's about the protein. And then another angle that I want to put across here before we talk about potential solutions is there's protein consumed and there's protein absorbed, okay? So animal source proteins, if you think of the perfect food, which as Betsy would appreciate, has undergone lots of evolution, is mammals' milk. And this is an example of something where all the protein in there is absorbed, and absorbed readily. And if you think about plants, even though the teparibrine's a nice plant, it still isn't, its absorption rate's only about 70%. So in other words, just the configuration of those proteins, how they're protected with anti-nutrients. So if we have this challenge where we need more high-quality protein, and what would we do about it? Naturally, as a nutritionist, we would say, well, these people need to eat more animal source foods, need to eat milk and beef and so forth, eggs. But that doesn't jive with the environment. So here we have on this picture, let's try it on this side. These are greenhouse gases. The longer the bar, the more greenhouse gases we got going. And these are foods. And so you see beef, lamb, beef from another kind of beef, shrimp, cheese. These are all the big greenhouse gas producers. To make these foods costs lots more greenhouse gases. And then you see dairy down here, and then you see plants. So people probably have heard of the Lancet Eat for the Future diet. This is all about plants, which doesn't necessarily meet the nutritional requirements of humans. It's not really managed that way. And the environment's not just about greenhouse gases. I mean, the environment's about water, as folks from Arizona, I'm sure, pretty know. And agriculture uses up 70% of the world's water. And particularly animal agriculture uses up lots of water. And then land use. So in the agriculture spectrum, 80% of agricultural land is used for livestock. Okay, so we're showing you here that even though to meet this nutritional need across the world, we would think of animal source foods, but then we might not think of animal source foods when we look at the impact on the environment. So I want to tell you very briefly about two considerable alternate. These are not experimental forms of protein. These are things that are consumed in other places in the world. But if we started to produce more of them or consume more of them, we might find some tremendous advantages. So spirulina is a bacteria that grows in brackish water. And really the most beautiful thing about spirulina is it doesn't need any fuel. It just takes sunlight and CO2 and it makes a plant. And that plant is 60% protein dry weight. So most other plants are, when we pick the right part of the plant, like a seed, which is usually the most dense part, we might get 25% or something. But here we have something that take the whole plant and you get 60, 70%. And when you look at we say, well, what kind of protein is that? You were telling me it needed 20 different pieces of building blocks. It's got all the right stuff in it. You can see the numbers over on the right side are going to be all higher than the numbers on the left side, and that means that's a complete protein, and it's very, very providing exactly what the human needs. It is already consumed in Asia by lots of people. It makes no greenhouse gases, so this is a candidate for a solution for protein malnutrition. Now, here, though, I want to say, so if it's so great, why don't we all just do it and then it'll be done, right? But it's not. It's got some challenges. It doesn't just grow anywhere. It doesn't grow in the Arctic. It doesn't grow in temperate ocean climates very well. But it needs certain wavelength of light and a certain temperature of the water. And the brackish water is not everywhere in the world either. Okay, so you may hear later about other ways that spirulina may be grown, but it's limited. We have to get around the habitat that it needs, and explore what kind of species differences there are within the cyanobacteria. Which ones could be more suited for different environments? Which ones could be more suited for different growing? Most of this slide over here is just trying to tell you that spirulina has a lot of bioactive compounds. So when I use that certain paradigm of how the human digestive system works, the macronutrients, of course, are carbohydrates, fats, and protein. But then there's a fourth group of small molecules that have their own active effect on health. And spirulina has a lot of these, and some of them are active on our neurosystem. The second alternative protein I want to mention to you is mycoprotein. This is a fungus, and societies have been eating this for thousands of years in fermented foods. It's yeast, and when it's dry, it's about 40-50% protein, which is huge. So when you think about many other plants, you're getting, corn is 5%, rice is 7%, get up to beans, they tend to be 20-25%. But these two things I'm mentioning here have much higher amounts of protein. And the neat thing about yeast is it doesn't need any sunlight. It grows in the dark. And all it needs is fuel. It needs, some kind of carbohydrate source in the water, and then it's fully capable as an organism of taking that and making all the different biological molecules it needs. So, and it's pretty quick. It can be turning around in about 48 hours. You can make a pond as big as half of this room full of yeast. I did it before, even decades ago, where in a water effluent from a sugar mill, and then we were using that as a food for animals. Both spirulina and mycoprotein are 100% digestible. All the protein that goes and consumed is absorbed, and again, the building blocks that are in short supply are in big supply in mycoproteins. So what's the deal with mycoproteins? What are the challenges we've got? The challenges have to do with developing this value chain of how it's grown. I mean, we don't find mycoproteins naturally. We find spirulina a bit naturally, which gives us clues. But developing the value chain, if we tried to supply all the world's protein for mycoprotein, we would use all the cement made in the world making cement ponds for 3 years. Okay, so this is not like, it is possible to make cement ponds, and that's how you grow it, because and it uses a lot of water. That's the deal with it. It uses water. It doesn't need sunlight, but it uses water. So all of these steps here of the value chain are areas that need development. They need work. They need resources to make this a viable alternative. So, in summary, I want to say a few different things. One is that there's not one answer to this problem of how we're going to feed the world and mitigate climate change. That's good, because we're going to need a lot of options in a lot of places. And what I'm here to tell you today is there are some options that are not plants and not animals. And those are not in our imagination. They're currently being used in different places in the world. And how we cultivate that and work that Arizona can be a laboratory for that. So anyway, now what do? I go and turn it over to somebody else, right? You?
Yeah, me. Thank you so much, Mark. That was wonderful, and I love the fact that you're talking about all different ways to attack this problem. It's not a one-size-fits-all, and we're talking about biofortification of crops, and we're talking about microbes to enhance the properties of crops, and now we're talking about other sources of protein and microbial sources of protein. So we can do this. We do have the solutions. It's a multi-pronged, but we do have the solutions. I know we normally say last but not least, but I don't know. With these speakers, this is a hard act to follow. But I'm next, and I want to speak today about vertical integration necessary to develop spirulina as a means for tackling global malnutrition. Whoops. I'm dyslexic. I've got it upside down. Well, there's a lot of slides that Mark has already spoken to that I really don't need to go over again. Spirulina is considered one of the most perfect foods to combat malnutrition. As Mark says, 60 to 70% of its dry weight is protein. Nothing touches that. It's rich in vitamins, filled with minerals, powerful antioxidants. So overall, spirulina is a nutrient-dense food that is perfectly suited to combat malnutrition. I remember the first time I began working with cyanobacteria, I went outside and my swimming pool was filled with blue and green algae, and I went, damn, that's some biomass. So it occurred to me that this is some biomass. This thing can make biomass, and it can make it rapidly. Spirulina is interesting right now because it may be the hottest superfood in the U.S. I looked up the number of companies, and there are over 30 companies right now, startup companies, matured companies, that are trying to make spirulina-based health foods. So it's a superfood in the developed world, and it's a potential panacea for the developing world for all the reasons that Mark just said and I just said. However, the thing about the developing world is there's just a handful of companies, and again, they're not well funded, and the vertical integration makes widespread application of this impossible. So, if it is indeed a panacea for malnutrition, why hasn't it been done? And again, part of it comes down to cost, and we're going to speak directly to that, because in addition to having great people in agriculture, we've got probably at the U of A the best builder of bioreactors for cyanobacteria in the world, designing. So Joel Coelho in the back. So the high cost, it could be reduced by 25%. Other barriers to entry is poorly optimized drying processes, strong variation in the quality of products, limiting demand for food products. Importantly, and very importantly, and Mark has left the room, but I'm sure he'll be back, there are no randomized, there's 4 randomized clinical trials that you can find in the scientific literature. And they're all, with all due respect, very poorly done. So if we're going to proof principle here, we need randomized controlled clinical trials. And again, there's been some interest in spirulina by charitable groups, but again, these have been poorly funded and clearly not vertically integrated. So I'm in the midst of my 5th startup and another nonprofit. And what I realized a long time ago is innovation is one thing, but I've already found out with the CNRS that multidisciplinary, transdisciplinary, bringing disciplines together is your focus. And it should be the focus, because perhaps the greatest innovation to bringing a product to the marketplace or solving malnutrition in Africa is vertical integration. That's the most important. So there are several steps with spirulina, and I'm going to go through them one by one, and we need partners. I'm glad the CNRS is here, and I'm glad others are here, because we need partners. We can't dance at every wedding, or we can dance but not well. So, whoops, can't dance at all if I do that. So vertical integration step 1 is the production of the biomass. Again, you can grow spirulina in open raceways. We're going to grow them in photobioreactors. They grow at pH 11, so nothing else grows with them. Once we grow, there are hybrid systems. Once we grow, growth parameters, and I'm not going to read all of these, but then harvesting them either by centrifugation and normal filtration, drying, grinding, pellet capsules. So let's talk about dramatically reducing the cost. Again, we're so fortunate to have Joel at the University of Arizona. I think he's patented 5 of these. So we're very fortunate to have him here. But they have to be simple in design and construction, and this is the main cost. They have to be excellent, stable mixing conditions, easily scalable. Again, we foresee this, as I said, we just got back from building a manufacturing plant in Ghana. We see this on the ground. We see this locally. So easily scalable, low cost, ready available materials, easy operation and maintenance. So Joel has come up with some prototypes, highly inexpensive prototypes, but highly productive prototypes. And these are air-based prototypes. You see on your left, a simple one. Again, Joel tells me that when you get to scale and you're simply pushing air up through this bioreactor, when the scale gets larger, the growth dies. So there's one that's more complicated, more expensive here on the right, but the one in the middle is sparging air up through the reactor. It has a center internal wall. It causes circulation, and I'm told by Joel that that would be the simplest and most inexpensive with the least expensive materials. And Joel actually has some pictures if you want to see this. He's actually produced it, and it's on top of our one of our parking decks at the U of A. So he's procured the materials and he's constructed the bioreactors. And again, he has the pictures. He showed them to me a few minutes ago. I'm so excited. I feel like I have another child. It really, 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 really nice. Again, now we just need growth trials, optimization. Again, focus on drying, focused on how we can make this scalable, how we can make this scalable in villages in Africa. So number 2 is to determine the nutritional composition of spirulina coming out of the prototypes in the drying process. My Center for Precision Nutrition and Wellness is very good at this. Again, we can look at ash, moisture, protein, carbohydrates, proteins, amino acid analysis, fatty acid analysis, micronutrient analysis, and then my lab in particular is very good at fatty acids, and as Tom Brenna in the corner will tell you, omega-3s are necessary for brain development, necessary during pregnancy. So fatty acids, and again, these are only bioavailable as certain lipids. So my lab can look at over 350 lipid species to see if we're producing the ones that can cross the blood-brain barrier and be bioavailable and enhance brain development. Number 3, production of spirulina-based food aid products. I think I'm most excited about this. Of all the things I'm excited about, I'm most excited about this. So there are peanut butter based, there are other based, milk based, there's other based ready to use therapeutic foods for severe malnutrition, ready to use food supplements for populations who are undernutrition but need more nutrition. And I think we have a great opportunity here to build new ready-to-use therapeutic foods and new ready-to-use food supplements. And again, 60% protein, adding the right fatty acids for brain development. So there's a huge opportunity, as I see it, for producing ready-to-use that will not necessarily compete, but have advantages over existing ready-to-use therapeutic foods and ready-to-use food supplements. Again, this is something that Tom and I have spent our lives on. This is one of Tom's slides, but one of the major advances that Tom and Mark have made was adding omega-3s to already existing ready-to-use therapeutic foods and ready-to-use food supplements, and the impact that that had on brain health, IQ, retinal health. Again, 10 to 15 clinical trials just by adding omega-3s to these ready-to-use therapeutic foods. So again, we will be making spirulina-based ready-to-use therapeutic foods. It already contains a unique fatty acid called gamma-linolenic acid. That is an omega-6 fatty acid that's the highest in the food supply, and we will be perfectly balancing that with omega-3 fatty acids. Step 3, again, these will be used in the developing world. Spirulina-based powder, spirulina-based energy bars, spirulina-based cookies, noodles, supplements. Sophia is my program manager. At least once a week, she is bringing me a spirulina-based food, and we're testing it. So we're already carrying this out in my stomach in my laboratory. So she's a gourmet cook, and she's done a wonderful job in producing some very, very delicious foods from spirulina. Clinical trials, as I said before, I think that there are only 4 clinical trials that I could find in PubMed that have looked at spirulina-based clinical trials since 2005. And if I might humbly say, they're very poorly done. They're not done to Mark Maneri standards. So we have the capacity with Mark to do these and do these extremely well. And in fact, Mark has already designed one clinical trial in mind. Again, in Malawi, again, it would be an individually randomized, investigator-blinded controlled clinical trial with a spirulina-based ready-to-use therapeutic foods with omega-3s to improve physical and cognitive recovery in children. We're already, and again, I won't go through the methodology, but again, these would be carried out in 10 locations in Malawi, 10 rural sites in Malawi. And again, this is a region where Mark has over 30 years of clinical trial experience. So if someone else was saying they were going to do this, you'd go, nah, they can't do this. It's too difficult. We can do this. Again, partnerships and global distribution. Again, the CNRS, we need your help. We need partnerships. We need your partnerships. Again, we need other organizations to play significant roles. But not only that, and Andy Moose was going to be here, and he's going to be here this evening. 5 minutes? Yes. He'll be here this evening. He had a sick child this morning, but he's the head of health and wellness for the World Economic Forum. These are the CEOs out of Geneva, Switzerland, around the world who really care. And so I just want to give an example of a nonprofit for-profit partnership. Mark and I, as I said, spent the last week in Ghana. There is a partnership that we formed with Nike and Adidas, and basically we took a crop shoe and we turned it into a high performance shoe. And right now with the pre-orders for this shoe, just pre-orders, we would be the 20th largest shoe company in the world, just with pre-orders. All that is being produced by the World Shoe Fund, Inc., the for-profit that drives the nonprofit that makes shoes for children throughout the world. So I'm on the executive board of the World Shoe Fund, but again, there's a for-profit driving the nonprofit, and again, driving the giving away of shoes to children throughout Africa and the rest of the world. I can see a similar model with spirulina-based medical foods and dietary supplements. I actually took a company public. I was a CEO of a company that took a medical food into the marketplace. It was called UArizona for asthma, allergic rhinitis, and psoriasis. It was a medical food. And I believe with the people in this room that we can come up with a very important either medical food or dietary supplement, hopefully medical food, because again, that's a different category. And I believe the for-profit sales for that could potentially drive sales for nonprofits. But I'm dreaming. I'm a dreamer. Don't fall in love with a dreamer. But I'm dreaming. So what are the potential competitors? We did an extensive examination, and it really, for this to be a panacea for the world, for malnutrition, it's quite pitiful, quite frankly, how little is being done and how the lack of vertical integration is being done. So there's good interest by a lot of small groups and underfunded NGOs. The one company, a Swiss foundation called Antenna, was the closest model, but they just went through a massive restructuring and laying off. We will soon need to clarify whether our vision is centrally or locally produced spirulina for a cheaper, more effective, utilizing cheaper, more effective bioreactors and other methods. And again, just coming from Africa and me personally spending the last 20 years in Africa, it would be my dream that every village would have a spirulina bioreactor farm in which they were taking spirulina and solving their malnutrition problems with spirulina at a local level or both. And I think that's it. Thank you. I think folks must have many questions, and I sure hope you do. And I'm going to ask the speakers to come up. I'm going to ask Tom. Tom is at the University of Texas, and he's going to join us. Joel's going to join us. Andy Moose was going to join us, but again, a sick child. But again, I'd love to bring the panel up and all the speakers. So Betsy, Duke, Joel. Okay, now's your chance. Go get them. Yes.
Great, thank you. I'm Ken Weiland. I'm the new director of the School of Nutritional Sciences and Wellness at Arizona. This is actually for Duke. You talked briefly, you had said, I know you don't have time to go through all the details in a small presentation here, but you talked about the tepary beans, if I'm saying that right, being plug and play, but then also we're not quite there. And there's a little bit of, can you talk more about what are the additional things that need to be done to bring this out more than it is now?
Sure, and I think that's probably one of the holdups of why this crop's not widely adapted is there is obviously a yield penalty. So it's hard for a traditional grower to be like, well, why do I want to grow something that doesn't produce as much? And by plug and play, I mean that you could take this bean plant and you could grow it in a traditional setting where beans are already being produced, no problems. Why I would say that we need time to get there is to make it probably yield more to basically drive people towards producing it. I think that would ultimately be what I would be getting at. It's tough. I mean, right, it's hard to convince growers to grow something that's less profitable, but at the same time, if you can grow it with less water, I mean, it does pencil out mathematically in terms of why it's beneficial.
Okay, Do we present ourselves? Maybe, because not
Yes,
everybody knows
please,
everybody. So my name is Martina Knoop. I'm the Director of Interdisciplinary Programs at CNRS. I had a question to Betsy about the what I understood from the what you told us about the symbiosis of microbiomes, which are plant microbiomes and soil microbiomes. Could you say something about the time scales? I mean, if we're talking about, I mean, you want to have this really perfect, efficient symbiosis between plants and the soil, but we see that in the changing world, soils are getting drier, and so the transition to arid land is maybe faster than what we plan to do. Yep. And so does the microbiome change with that, or do you really have to intervene there?
Yeah, thank you. That's a wonderful question. An aspect of microbiome science is that it's a study in dynamics, and we can see very rapid shifts in microbiome associations. One thing that we see is that as plants are moving from a well-watered condition over a short period of time to a very dry condition, there's a shift, for example, in the prevalence of filamentous bacteria or actinomycetes, and that can happen over the course of less than a single growing season. So we should think of these as an extension of the dynamic adaptations that a plant has, sort of its arsenal in real time. The challenge that we have then is to integrate those instantaneous or short-term changes into a longer-term framework for stability. And I think that's one of the hard parts for microbiome science going forward for sustainable agriculture. That is, how do we handle rapid shifts in microbiome associations in a longer time context? And that's where ecology and evolutionary biology help us, because we understand what the guardrails are on those short-term dynamics and how to establish those partnerships that serve over a longer time period. So I hope that answers your question.
You have to have an active,
Yeah.
You have to, I mean, if you want to have let's say a mean-term evolution, you'd still have to intervene actively in the process, and you cannot wait for it to happen spontaneously.
That's an interesting question. The shifts that we see for plants will happen as a spontaneous collection of soil microbes will be selectively filtered to enter into the plant. And so the question then is that if we're seeing these soils changing over time and they're losing biodiversity, as with legacy agriculture, that's where soil additions or inoculum may become especially useful. And that's why we need to know what's in the wild world around us before we're in a purely post-agricultural age. Thank you.
I'm Nathan Sherrington, and I'm not a plant pathologist, but the director of the Center for Toxicology. You mentioned the idea of the plant developing natural defenses with the microbiome. Is it a signal from the plant, or is it natural selection by the drought that causes the changes?
Yeah, a wonderful question. So if I can rephrase it to make sure that I'm understanding, the question is the microbiome selectively choosing to colonize under certain conditions, or is the plant filtering for a subset? Who's in charge, basically? And I would argue that in a symbiotic framework, both partners are communicating with one another, and that it's always a subset of what is locally available that will end up inside. The signaling that happens is often at a hormonal level between microbes and plants. Many microbes that are part of the microbiome do produce plant hormones, and that is a massive part of their signaling currency. But we also know that they can influence gene expression of the plant hosts and so on through other means. And so the who's in charge question, I think, is one that is perhaps best reframed as how do both parties in charge work together? And that's what's exciting to me. I think that frontier of recognizing that there's microbe communication, there's plant microbe communication, and then there's the abiotic filter that lays over all of it. So that's not a clear, simple answer, but one to say that that's a research frontier. And understanding theoretically, conceptually, as well as practically who's in charge is a great question.
I can ask a very French question, but you will see. What is so? I mean, taproot beans. I didn't know about taproot beans. So my question was, what's the taste of taproot beans? But when I mean, you will see. No, you will see what I mean. What I want to say also is what you said, Duke. You said that. I mean, if you change food habits, people have to. I mean, you said this is easy because people are already feeding on these sort of beans or on beans in a general way. So in introducing something which is not well known, there's also this part of how there's the uptake by society or by the people you want to feed with that, or by livestock. You said this is something that livestock can feed on. Will livestock feed on that? I mean, is there really are there studies about that?
Yeah, so in terms of using taproot as a forage crop, New Mexico State is actually leading the way on that because they're in a very similar crisis of not having even water to do alfalfa. In terms of taproot bean taste, it's very good. Most people actually prefer it because it actually, well, it's hard to travel with. I mean, TSA. It's very earthy. It has more flavor than like a common bean or a navy bean. Most people generally prefer it. In terms of like cultural adaptation, when I was a postdoc, the lab I was in, they were working with Harvest Plus and making biofortified corn. Gates Foundation had poured millions of dollars into this project to make biofortified corn. It was orange. So they released the corn variety. They came back in a year and a half. They went to the small towns and they said, "Where's all the corn?" And they had all piled up and they were feeding the pigs with it. And they said, "Why are you doing that?" And they're like, "Oh, people, we only eat white corn. That's the good corn." And they've taken all this biofortified corn. And that's the thing about like when you introduce food into society, you have to understand the cultural significance of food. And that's why with taproot bean, we're not asking them to grow anything different. We're just asking them to kind of grow a different style of bean. And that's kind of why another reason, like plug and play, there's no like, you don't have to get your mind around eating it. And that's one of the nice attractive things about it is it fits in nicely. I do think Martini would love the taste. It is really good.
So the University of Arizona is in Tucson, and Tucson is home to Tohono O'odham Nation, which is a Native American tribe, among other tribes. But the taproot bean is one of the staple for ages of this Native American tribe, and so in terms of its the history of it being part of the diet of humans, it's well established.
So I'm Jerome Rose. I'm working at the Earth and Space Division at the CNRS in charge of research infrastructure. And thank you very much for the great talk. My background is digital science, so maybe I'm coming from far, but I will learn a lot. I would like to make a first comment about what you mentioned about the need of interdisciplinary. I like your vertical integration of any innovative process is really the key. So I fully agree with that. And with that in mind, we also have to take into account that any innovative process has some weaknesses. You mentioned the challenges and maybe some risks that can be taken by this innovation. So that's also part of the process of the innovation. And I have a question about whether the plant of the microbiome, I don't know whether the question is for both, but when you think about, for instance, a plant that is native of a region and you want to use that plant in another region, but you have other native, do you think there are any risk that the plant can be aggressive to the native ecosystem, or is there any, or the microbiome you want to transfer, is there any research about this?
I'll comment after you. Okay, go ahead.
I can only obviously comment on the plants. Well, taproot bean has been grown in Africa and India and these other regions around the world, and they haven't had any real problems. And in fact, what is it, Akarta? It's the International Dryland Institute in Morocco. They're actually crossing taproot bean with common bean to try and break that yield barrier. And so that's another reason why it's really a good solution, is actually already being pursued at some level, although modest, globally. And so I think, if I may, to answer your question, no, there's not really a concern, and I do think it is broadly adaptable to other regions, and especially as we think about climate change, really the world's becoming a better environment for it. So I think that's one of the reasons to overcome that.
And just to follow on that, one rule that I think is emerging from microbiome science is that because there is variation in the available microbiomes around the world, an ideal innovation may be for a given country to use its own microbiome resources, and therefore to be developing a natural resource that is as good as gold. And with the training and infrastructure that go along with that, it's a prospect where having a few thousand microbes in culture may end up yielding the new medicine for that country that they never knew they had. So my vision personally is working at a regional scale and not necessarily exporting Arizona microbes, but instead helping select for the regional microbes that are important for the flourishing of that crop in that area, and thus are a national resource there.
I had a quick question for Tom. You and Mark have obviously done landmark work in this area, but Ski's a very enthusiastic guy, obviously, and I'd like you, based on the spirulina proposal and the vertical integration, to poke some holes, or what do you think the challenges or pitfalls are in that plan going forward?
How much time do I have?
Oh, heavens.
Well,
Don't hurt
first
him.
I want to thank you for having me. Thank you, Ski, for inviting me. This is a lovely event. I promised myself that as soon as I got my hands on the microphone, I would say that in most of the time when we're talking about agriculture and food, we're talking about nutrition below the neck. But what's really important, and the most important thing, is nutrition above the neck, particularly for mothers and babies. Try to keep that in mind. It's not just calories and protein. It's how we grow the best brains. And that often gets lost in the mix. We get tired because we've fixed the protein problem in a little place. Okay, so now the really special thing about the brain, you do need a lot of protein and you need iron, and I don't know about the absorption of iron in that stuff. And Mark mentioned the word anti-nutrient, which we don't use that word very much in the United States. They use it in Europe, but we don't use it much here, and that's an important issue to deal with as well. So those are a couple of things. The thing that's really special about the brain is the fat in the brain. So the polyunsaturates are really highly unsaturated fatty acids, and there's lots of nuance around this that led us to start working on this maybe almost a decade ago and continue to work on it. Yes, we need protein to grow a brain. We also need the right fats, and we need the right mix of fats. And that's one of the things I was wondering about when I was looking at that. The beans here, and I'm a little disappointed we don't have a crock pot out there of, because what the heck. But anyway, the little bit of fat in there looks okay to me. It looks a little like canola-like, actually, but since it's low, that's good. All that's good. He already knows this. So that's okay. But there has to be something else in the mix. It's not gonna. I don't think it's gonna be all that kind of beans that does it. We've got to grow. We've got to grow brains with the right kinds of fat. Now, you asked me about vertical integration, and vertical integration I don't think is a very bounded thing, right? So it's almost like a permission to talk about anything. That's why I went to the fat immediately, and there's all those issues in the cultural thing. Actually, I'm glad that was brought up. So I spent most of my academic career at, as I like to say, a cow college in upstate New York, Cornell, and the nutrition department there. Some of my best friends were folks who would have a look at something and say nobody's going to eat that because it's not a big enough thing. They don't eat little fish; they only eat big fish, and they fillet it like this, and they throw out the best bits, and so you really have to take care of that right up front, like that orange corn that they feed to the pigs, which our pigs are often much better nourished than our mothers and babies, unfortunately. So so I've hit the cultural thing, and what else? I mean, God, I agree with Ski that that you almost start out with a fantasy, and then you start knocking things out, and if you get really lucky, you live long enough to see it to the end, right? So there's a million things to solve. So I once on the other side of town, I spoke for 3 hours and 20 minutes without a break. So I'm gonna stop there. Okay.
I want to go to Joel a second because Joel has been so important in this process. And one of the questions I'm often asked is, if this is such a good idea, and, NASA, WHO, everyone says this spirulina is the food of the future, then what is the issue? Why are we not growing it everywhere? Why were we not doing? What is the major limitation from a production perspective, and why is the cost still somewhat high, and how can we address that?
Right. Thank you, Ski. That's a crucial question. So my background, I'm a professor of agricultural and biosystems engineering at the University of Arizona. I work with vertical farming on one side, and I work with bioreactor design of various cell cultures. So I've worked with all types of cultures. And that question, really the answer to that goes back to the fact that crop agriculture has been around for 10,000, 12,000 years. But when it comes to culturing photosynthetic microbes, like, plant cells, as well as spirulina and algae cells, they were only developed about 1939. So, for instance, in Texcoco, Lake Texcoco in Mexico, the Mexicans or the Aztecs, before the Spaniards came, they had spirulina as a staple diet from the lake. But the lake just happened to have the right conditions to naturally grow spirulina. They were not farming it. They didn't know how to farm it, but they were just harvesting them, and that became part of their diet. Now, unfortunately, when Mexico City was built, part of Lake Texcoco was drained, and so they lost the spirulina. But anyway, fast forward to 1939, that's when scientists were able to start growing in vitro, in bioreactors, plant cell cultures, as well as algae cultures that are photosynthetic. But as you know, this requires specialized skills. So agriculture, when it comes to macro crops growing in the field, that's relatively easy, to impart to, from generation to generation. But in terms of intentionally, deliberately mass-producing microscopic algae and cyanobacteria, that requires specialized knowledge. So that really is the bottom line.
So I've told I'm got the 5-minute point, but I want to follow up on that in just a second. So what the royal you are doing is you're producing high productivity, low cost bioreactors. And again, I've seen the materials. The materials are basically things that you would find in a village. And so tell me a little bit about that, because that seems to be an incredibly important step in reducing the cost of spirulina.
Right. So to mass produce the algae, and we need to mass produce the algae if we are going to really turn this into a solution to be able to feed many, many people. And the way to go about that is to grow them in bioreactors. To grow them in open raceways, that's really very risky because of a lot of pollutants and contaminants and experiences of culture crash. But when you grow them in bioreactors, you have more control of the growing environment, and so the productivity is higher and more reliable and consistent. And I have to say, and I'm sure probably many of you are aware, there now is a global industry on algae production. So, for instance, it was mentioned earlier about the need for certain fats for brain development. So you're talking about DHA, EPA. Fortunately, there are microalgae that produce this naturally. And those microorganisms, those microalgae, are being grown in bioreactors commercially. For commercial production. And so the technology is there now. The key is to come up with a production system, a bioreactor, that is low cost, that is easy to maintain, that's easy to construct, that is not too sophisticated, that regular folks in villages in the global south can easily learn how to build them and to maintain them and operate them. That is the key.
And I think it's worth noting that Joel is working with several governments. I can't find him on the U of A campus because he's in Saudi Arabia, he's in Central America, he's teaching in the Philippines vertical farming. I know all over the world cheap vertical farming. So this is not his first rodeo. The other thing I would say about spirulina is it grows at pH 11. Mm-hmm. That's an incredible advantage because nothing else grows at pH 11. So you keep a very, very sterile and not so sophisticated bioreactors. Yes. Given that. I think we have one more question, perhaps.
Yeah. Joel, thanks for bringing up the scaling up of this, because I think we always face that when we go from bench scale to real world and trying to solve mega problems. So my question, and I'm Mark Verhoekstreet from the College of Public Health. I'm not a plant scientist, so I'm assuming, though, that plants do take up chemicals or pesticides from the environment that they're grown in. And so until we get to this highly controlled vertical farming in bioreactors, how do we control the environment to make sure we're minimizing the number of pesticides or other chemicals, man-made chemicals, in the final product, plus also making sure that no other algae that has the potential to grow like other toxins or produce toxins is included in that final product?
Right. So that's a great question. And that's the main advantage of growing the microalgae cultures in bioreactors, no matter how rudimentary they may be, as opposed to open raceways. Because when you have open raceways, it's open to the environment, and a lot of contaminants, including from the air, could end up in the culture. But when you have a bioreactor, it's pretty much a closed system. So you regulate what's going on inside, and so it's harder for environmental contaminants to get into your culture.
And
I'm not
Mark,
saying that there's no possibility for contamination, but it's greatly reduced.
And Mark, to your question, most spirulina are toxic. I mean, I just genetically engineered some to make omega-3s. So to find non-toxic cyanobacteria species are difficult. Spirulina just happens to be a perfect one. But even then, we're going to make sure before we give it to babies in clinical trials, we're going to do a high bunch of toxicology studies to make sure they contain nothing that could be detrimental. But again, Amazon has them in 10 products right now. So
I can aspirational comment here. We're trying to make our plastics so they're biodegradable. Why don't we try to make our pesticides the same way?
Can I just have one comment, please?
Yes.
So in regard to the deployment and implementation of this technology in villages in the global south, it would be really nice if that could be paired as well with the organizations of the growers and the farmers. And that is if they can be organized into not just villages, but cooperatives and farmers associations, so that economy of scale could be realized. And one advantage of that as well is in terms of facilitating the training of how to really mass produce or scale up the operation.
we have some inns there, some really important inns in Africa. this factory that we just produced, there's a massive farmers group that we have access to. So I think that's a part of the vertical integration that we'll be
Right.
able to pull off.
Thank you.
Well, thank y'all, as I would say in the South. Thank you all. I really appreciate you all, and I appreciate the questions, and I appreciate your attention. And again, thank you so much. Thank you for joining us.